Good afternoon. Thank you so much for inviting me to kick off this afternoon session. And unlike the song you just heard, I'm not blue, actually. I'm very happy to be talking to you today about blueberries. So I am your plant representation today. So before we begin, I want to tell you a little bit about myself. I'm in a unique position as a USDA NIFA postdoctoral fellow. I'm actually finishing that up right now at Hudson Alpha Institute of Biotechnology down in Huntsville, Alabama. But I'm also the co-founder of Veil Genomics. And the work I'm going to show you today is a reflection of that intersection of all the data that I'm showing you was generated in the academic lab with my fellowship, but it's exactly what spawned Veil Genomics and what we do there. I also want to go ahead and thank Dr. Juliet Chu. She is the blueberry breeder at University of Georgia, and she helped me put together a project that is building a custom pangraph for her specific mapping population of blueberries and using that to trait map blueberries. So today, first, I'm going to talk a little bit about blueberry background, so we're all on the same page about this wonderful fruit. And then I will go into HiFi for genomics, or for building genomes. And then HiFi for genotyping. This is really the meat of the project, or of the talk today. So blueberries are a really interesting crop for many reasons, but one of the reasons is that they're one of the only crops we eat a lot of that is actually native to the United States. And here I'm showing you a map of the top blueberry producing states in the US, and you can see there's a wide variety of regions where this is grown. And because of this, we have to have different types and also different species even of blueberries that we grow. So up north where it's cold, we grow Northern Highbush, and they need a lot of chilling hours to produce fruit. Down in the south, we produce Southern Highbush and Rabbiteye. And so I mentioned I'm in Alabama, working with the University of Georgia. So my project focuses on Southern Highbush and Rabbiteye blueberries. A little bit more about these two blueberry types. Southern Highbush is what we mainly produce down in the south. They are really good fruit quality, and that's mostly why they're the favorite of farmers. And they're also they were purposefully bred to be in the southeast, so they're an interspecific hybrid. Rabbiteye is a little bit more interesting. Rabbiteye is actually native to the southeast, and you can see from this picture it's a lot taller. It's more vigorous, it's more tolerant of the heat and humidity we have in the south, which is why we're interested in it. But the fruit quality isn't quite as good. But if you live in the southeast and you're looking for something to plant in your yard, I would recommend Rabbiteye. You won't really have to worry about it. And because we are now talking about plants, they have very weird genomes. So Southern Highbush blueberries are an autotetraploid, and Rabbiteye is a hexaploid, so 6 copies of the genome. So just keep that in mind going forward because these guys get kind of complicated quickly. My project focuses on a few traits of interest, primarily phenological traits of days of flowering and days of fruit set. I also work on fruit weight because I imagine we can all agree that we all like larger blueberries. So these traits are actually super important everywhere, but particularly in the southeast. Days of flowering is essential because if we have a blueberry that flowers too early and then a late freeze comes through, it will kill those flowers on the bush if they're already flowered, and then we won't have any crop that year. And that really seriously impacts our growers. And this is something that we see in the real world all the time. Similarly, we are looking at days of fruit. So how long it takes to go from that flower to having a fruit that we're going to pick off the bush. And if you've ever been to the southeast in the spring or summer, you know it is hot and it is humid. And so the more time these berries live in that environment, the more likely they are to have issues with skin softening, splitting, and rot. So we really want to minimize how long they are on that bush. And so we want to identify molecular trait or molecular markers that we can use for these traits. And just to harp on why these molecular tools are so important in blueberry, blueberry is a perennial crop that has a really long juvenile stage. So if you're trying to identify like fruit size and you have two parents and you think that across from them is going to have a really good blueberry with good big fruit, when you plant that seed of that cross in the ground, it's going to take at least 3 years until you can even start phenotyping the blueberries, and 5 or 6 years until you're at full productivity of berry production. And so I personally am way too impatient to wait 6 years to see if my berries are good. So if we can have molecular markers, then we can just wait until that seedling has just enough tissue to take DNA, and then we can select for it and move forward with our breeding program. So now that you guys know just a little bit about blueberries, I'm going to talk about building genomes. So part of my project was first building one reference genome for Southern Highbush blueberries. There are some other reference genomes for other types of blueberries, but not for Southern Highbush. And Susie Blue is the one that we chose, I think mostly because she has a really cute name, but also because it's a really good cultivar out of UGA and is a founder line to my mapping population. And long story short, with the wonderful HiFi data we got from our PacBio Revio, we were able to assemble the genome. One thing I want to point out real quick is the Contig N50. It's 12 megabases here, and that might not seem that exciting to you, but to me, that's actually really exciting, because if you look at older iterations of any species in the blueberry family and their reference genomes, most of them tend to still be in the KB range, probably due to how difficult these genomes are to decipher. And so we have a really contiguous genome, and through HiFi data and Omni-C data, I was able to do a lot of manual curation in the HiCMap and actually assemble and phase this genome. And thank you to Zach Maharg from the Harkis Lab at Hudson Alpha, who kind of showed me the art of doing the puzzle solving of HiCMap manual curation. I mentioned before as well about wanting to build a pangraph, and I sequenced 5 other Southern Highbush lines and built Contig level assemblies, but I didn't have the money or the time to do Omni-C scaffolding for all of these genomes. So I wanted to do reference-based scaffolding, where we map it to my reference genome and decide where everything goes based on that. The tools that were readily available for reference-based scaffolding really struggled with the auto-polyploid nature of the blueberry and made very wonky decisions that I was unhappy with. So I need to shout out one more graduate student from the Harkis Lab at Hudson Alpha, Laramia Kozbeck. She built a tool called Yet Another Reference-Based Scaffolding Tool that does alignment for you with Minimap and makes some, it has some idea of where things will go. But then you can go in and actually manually move it around. So you go from a genome that looks like this in the alignment to this. And so that is the way I was able to put together a relatively complete phased genome for the 5 other genomes. I then used an open source tool called Kufu Pan to actually build the genome. It's basically a wrapper of other tools such as VG and minigraph cactus, and it builds the pangenome, maps, and calls variants for you. And this is out of the brain of Dr. Walid Karani. He was kind of the architect of a lot of the work we do at Hudson Alpha in the Clevenger lab. And he has since moved over to Veil Genomics and is now using his brain to put together a suite of analytics that are specific to what we do at Veil, which I'll talk about more at the end. And just a quick aside about Rabbiteye, because I talked about it before, I also had a plan to build a reference genome for Rabbiteye, and we followed the same technique where we did HiFi data, short read Omni-C, but no matter how many hours of trying to do that puzzling with the HiCMap curation, I was not able to put together a fully complete genome. And that is when Jake from PacBio, not State Farm, asked me if I wanted to do CiFi in this blueberry. And if you haven't heard of CiFi yet, It is long-read chromosome capture. And this came out of the dentist lab. So I adapted that to work with Omni-C. And because I had already done the short read work, I could directly compare the results from my scaffolding work with the short read Omni-C to that long read CiFi data. Okay, so to show you the results of that, it also took a lot of work and manual curation. But what we see here is on the standard workflow with the short reads, this is a dot plot of the best 12 chromosomes that came out of that assembly mapped to my Susie Blue HAP1 reference genome. And what you can see here is there's a lot of gaps. Even with all the effort I put into it, the standard just had a lot of gaps and a lot of truncation, particularly at the ends of these chromosomes. Now, with the CiFi data, which I want to point out is only 4x coverage compared to that Omni-C 41x coverage, and only about 1.5 kb in length, I was actually able to resolve those chromosomes and put together a primary reference genome for the Rabbiteye. So that's very exciting for the blueberry world. So now that we've talked about building these genomes, now we need to talk about how we're going to use them and the genotyping. So now I'm going to talk about kind of the bulk of what I wanted to discuss with you all today. So I am at a PacBio conference, so I imagine most of you are very familiar with how sequencing works on the Review, but if not, I'm just going to back up for a minute. So in the plant world, we have typically used a Revio SMRT Cell for just building reference genomes. So if we have a Revio SMRT Cell, and let's say it can give us 100 gigs, I know it can give us more now, but my brain needs even numbers for math. We would put one single sample on that Revio cell. So for blueberry, which is a 2.4 gig genome, it would be about 41x. And that's how we would build that genome. And we still do that. But in this age of having a lot more reference genomes available to us, we wanted to start thinking about how else we could use HiFi data. And so we started thinking about pooling multiple samples together and using it for long read, low pass sequencing. And while the concept is simple and execution is a little bit more difficult, we knew that the wet lab work to do a scalable high molecular weight DNA extraction, particularly in plants, was going to take some elbow grease. Plants have a pesky cell wall that makes it kind of difficult to get that high molecular weight. So I was waiting for my USDA fellowship to kick in 2023. And so I'm going to take a detour and talk about peanuts for a minute. I didn't mention before, but the Clevenger lab is a peanut breeding lab. So me working on blueberries, I don't really consider myself a black sheep, but maybe like a blue sheep of the family. So I went over to the dark side of peanuts for a minute, and peanuts, because they are also a plant, they have a weird genome. So they are an allotetraploid, so 4 copies of 2 different genomes. And the first thing we wanted to look at what long read low pass might look like in silico, and particularly how it affects what we call confidence of alignment. So when we do a lot of genotyping work, we map, of course, the reads to our reference genome. And short reads, being only about 150 base pairs, are not that unique, so they tend to map to multiple locations, and then we have to filter those out. But long reads, being a lot more unique, are able to map a lot more uniquely, and then we end up filtering less out. So you can see here in this graph of this preliminary data that the short reads, we ended up filtering out about 50% of our data. Whereas with the long reads, by the time we're at 13 kb, we're keeping almost 93% of our data. And this is really essential in the plant world too, because we really want to keep all of our data. We tend to not really know where in the genome our trait of interest might be correlated with or causative. So we need to keep all the data we can. So then we did the work of actually figuring out that wet lab pipeline and how to do high throughput, plate-based, high molecular weight DNA extraction and library prep. And we did this work on 130 peanut lines. So now we have a whole small population that we were able to do this work on. And as you can see from this box plot, we looked at confidence alignment of alignment again, and we see what we saw before holds true with real life data. So the peanuts with the long read low pass are above 90% retention rate, whereas the short is still in that 50 to 60 retention rate. But I also wanted to look at whole genome coverage versus depth. So we often talk about depth, but depth just refers to the average number of times a given nucleotide position in the genome is sequenced. So it's just like redundancy at a specific base. Whereas coverage refers to the proportion of the reference genome that is actually sequenced. And so what we see in short reads is while it might say 1x, 2x depth, there tends to be large swaths of the genome that actually don't have any coverage because those short reads tend to stack up, particularly if you're doing a PCR-based library prep. So we looked at this with our peanut data, and we looked at it on whole genome as well as just gene-space coverage with the filtered and unfiltered data for that misalignment. And what you can see here clearly is even at the same depth, the long reads have much better whole genome coverage. So once again, I want to harp that we often don't know what we're looking for in plants or where we're looking for it. So trying to find that QTL that's correlated with our trait of interest is kind of like trying to find a needle in a haystack. And if we're tossing out half the haystack, then we're kind of shooting ourselves in the foot. So having all the data at our fingertips is really essential in our work. And to translate that into actual variants detected, I looked at long read versus short read SNPs, indels, which I consider 2 to 50 base pairs, and then structural variants, so anything over 50 base pairs. And we see, of course, that the long reads just kind of blow the short reads out of the water with variant detection. And just to take a moment to talk about structural variants a little bit more, it's very hard to call structural variants with short read data alone. So I wanted to see how much of an impact these SVs might actually have. So I put all this data through a tool called SNP-F, which looks at how these different variants might impact genes. And we see that of all the SVs called, almost 14% of them were considered high impact. So they're disrupting a gene in some sort of real way. Whereas the indels and SNPs are less than 1% high impact. So being able to call these SVs is very important. And to give you a real life example of this in peanuts, if you were at PRISM in San Francisco last year, my PI, Dr. Josh Clevenger, talked about tomato spotted wilt virus resistance. It's difficult to genotype because it's a large insertion consisting of gene repeats where more genes means greater resistance. So short reads, even on a pangraph, make this really, really difficult to genotype. So we looked at this with the long read data versus the short read data, and I want to show you via this IGV image where I have it mapping to the region in the donor parent that has this insertion. And what you can see here is we have our negative control and our positive control. And then we have a query sample that we thought did probably have tomato spotted wilt virus, and it maps pretty cleanly to the donor parent here, whereas the short read version of that same exact sample totally misses it. So if you know you have an SV that you need to be able to genotype, you have to have long reads. Okay, I know you guys were probably like, when are we going to get back to blueberries? So now we're back to blueberries. In the meantime, that I had been doing all this long read work with peanuts, I had been doing short read skim sequencing with blueberries because that's what we had written into my project proposal. But having all this data with the peanut made me think I really want to do long read sequencing with blueberries. But I didn't have the funding to do 600 samples, which is my mapping population size. So instead, what we did is we pulled out the samples that were the extreme phenotypes from the population and did a bulk segregant analysis type experiment. So in the end, I did long read low pass sequencing on 96 blueberries, targeting around 1 to 2x. And I had short read data to compare as well. So I mapped both of these to my previously constructed pangenome. And what you can see is, like in the peanuts, the long read low pass data just blows the variant count out of the water compared to the short reads. It identified 16x more variants overall. And those variants did actually mean something. When we look at the potential QTL regions detected of those variants, it's 59 versus 22. And so for me, that's more than twice as many regions I can explore that might actually be really important for uncovering the genetic architecture of my traits of interest. So an example of that is with fruit weight. Fruit weight I didn't talk too much about before, but it is a trait that we care a lot about, but it's difficult to elucidate. There's a lot of factors that are involved with fruit weight, making a berry big or small. It's highly quantitative. But with the long-read low-pass data in this BSA experiment, I was able to identify a region on chromosome 4 that has a lot of markers clustering for fruit weight. And the top marker here even has a percent variance explained of 17.3%, which is quite high. And when you look at the actual breakdown of the allele effect there, you can see clearly that a higher dosage of the alt allele leads to a larger berry compared to the lower dose. And if you look at the bottom here, looking at the short read low pass on the pangenome, it totally misses this region. So this is something that we would be out of luck with if we didn't have the long read data. And while there were 59 regions identified, I'm not going to show you guys all 59. You would probably get bored. But I wanted to pick out another couple to show you. Here's an interesting one at days of flowering that has a pretty high PVE. And as you can clearly see, higher dosage means later flowering time. So that's what we want, a high dosage of the alt allele. If we look at days to fruit set, we see that the lower dosage means earlier fruiting. So we want a combination here that is the late flowering, early fruiting. And to show you what that might look like if we could actually combine those samples, and I actually do have some that have this allele combination, we can look at it in terms of dates that we can that's like more translatable to real life. So in our unfavorable allele combo, the blueberries on average would flower on February 20th. And in Allahapa, Georgia, which is where these blueberries are grown, the chance of a freeze event after February 20th is still 70%. Similarly, if they fruit on May 8th, the temperature in Allahapa is already at 84 degrees, so it's starting to get a little steamy. But if we have this favorable allele combo, we can push flowering time to March 12th. And after March 12th, there's only about a 20% chance of a freeze event. And then for fruiting, if we fruit on April 24th, then our temperature is now only 78 degrees. So you can see how the results we're finding here actually have real-world implications for our farmers. And that's how we go from, and we also can go from 77 days, like on the bush, this growing time, to only 43 days. So we can greatly shorten that production time. And now I just want to end by talking about Veil for a second, Veil Genomics. We are a startup company that is an associate company of Hudson Alpha. So we are also based in Huntsville, Alabama, in the Hudson Alpha building. And we are up and running and operational. It was founded by real scientists, myself, Dr. Josh Clevenger, and Dr. Alex Harkis, who are faculty members and experts in their field at Hudson Alpha. Both of them actually spoke at the different PRISM events last year. Hopefully you got to see some of those. And what we do is really offer a streamlined wet lab procedure. So right now we're mostly a services company. We are able to get you from tissue all the way to sequence data. And not just in plants, we're doing this in animals, microbes. We've tested all this as well. And we also work with our customers really closely for consulting with design of experiment. We talk to you, we'll do pilot studies. If you aren't sure you want to do 1x versus 5x, we can help you figure that out based on the goals of your specific project. And we're also very soon launching our Veil Analytics platform. So very soon we'll be able to give you analytics as well. Everything I showed you today was done with just open source tools. So already there's a lot that you can do with long read low pass. But Dr. Waleed Karani, who I mentioned before, is a genius computational biologist, and he's working to build out a platform that's actually specific to long read low pass data. And we are very excited to be partnering with a unnamed big ag company and big tech company to do some really exciting work that I think will revolutionize the way that we can handle large data sets. So please follow Veil Genomics on LinkedIn or on your socials for further announcements that might come about. Reach out to us if you think you might have a project that fits this kind of workflow. And with that, thank you so much for listening to me talk today. I want to thank my lab mates and all the people who helped me put together these genomes because they were very difficult, as well as members of the Harcus lab. And unfortunately, I really just talked the whole time. So I think that I don't have time for questions, but I will be around the rest of the evening if anyone has anything they'd like to talk about. Thank you so much.